Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Dr. Chantal Abergel. She's at the CNRS Research Center. She completed a Ph.D. in material science in 1990 at a university that I probably can't pronounce offhand in France. She was co-founder of the Structural and Genomic Information Laboratory at the CNRS with Jean-Michel Claverie to create the first French lab that combines bioinformatics with experimental biology, which makes a lot of sense. And now she's isolating and studying families of giant viruses. And even though they're giants, I'm sure they're pretty small, but we'll talk about the work there. So, Chantal, thanks for coming.
It's coming fine. It's perfect.
Well, good. Yeah, well, tell me, first of all, how big are viruses in general, and how big is a giant virus?
Oof, a tough question. I mean, in general, viruses were thought to be very small. Historically, they were the first virus that was isolated by a scientist who wanted to find out what was causing a disease in tobacco plants. And surprisingly, he filtered the sample he extracted from the plant. And instead of isolating it on the filter, it appeared that this agent, pathogen, was able to infect other plants while it went through the filter. So the first one was very small, passing through a 200 nanometer filter.
Oh wow! Now, and was that tobacco mosaic virus?
Exactly, and that was actually the first that was discovered. And because it was isolated that way, it became traditional to isolate viruses using exactly the same approach, meaning filtering. So that way, there is no chances to get in contact with giant viruses because you always look at the fractions that pass through the filters. And those giant viruses are so big that they are actually retained by the filter.
Yeah, I've heard that a lot of viruses, in the virion stage are 50 to 120 nanometers. Is that accurate? And then how big are giant viruses, if so?
I mean, the largest can go as high as 2 micrometer. So it's much bigger than the standard viruses that were isolated previously.
Wow. How much genetic material do giant viruses have? If they're so big, do they have tons more genetic material?
Yeah, they are as complex as they are big. They are complex on every direction. The point is today, we're only isolated DNA viruses. Giant viruses are all DNA viruses. But their genome can go from a few hundreds of kilobases to about more than 2.5 million base pair. So that means that they can have as many genes as even eukaryotic cells. So it's for the largest, it's about 2,000 genes. So making 2,000 proteins.
Yeah, I think Mimi virus is one of the giant viruses. Is that right?
Yeah, that was the first. Mimi virus was the first to be discovered. And fortunately, it was an icosahedral virus. So because of that, during the isolation process, it appeared immediately that it could be a virus, but a giant virus. For in the case of the Pandora viruses, which are very different in terms of particles shape, they are ovoid. It was much more complicated to be certain that they were giant viruses as well.
Oh, because they would be mistaken for bacteria or protozoan or something else?
Exactly.
Do they have any motility, or are they just free-floating? Have they been observed in solution?
And they're not even free-floating because they are so dense that they actually sediment. So no motility.
No motility at all, okay. But because they don't have that, traditional icosahedral head or, shape to their capsid, they would be much harder to recognize as a virus.
Yeah, actually, they were spotted by a group in Germany who reported the parasite of Acanthamoeba cells, amoeba. But they thought that it was more like an archaebacteria or something like that. This is only when we studied the infectious cycle that we convinced ourselves that there were Pandora viruses. And to study viruses, I think we should be as open-minded as possible because they break many dogma.
That's true. Well, in the interest of being open-minded as possible, do you think that viruses are alive, or at least contingently alive, once they enter into a cell?
Okay, the point is I do think that they contribute to the life. It is definitive, but I also think they are alive. They are part of the evolution. They are part of the life on Earth, and they are very active, actively co-evolving with the cells.
So why giant viruses? What do these viruses tend to do? Why are you interested in them?
Why? Because they are fascinating. That's the first answer, I would say. And also because they have so many components for which we have no story at all. They are made mostly of genes of an unfunction. So studying them is a way to prospect new metabolic pathways and address questions such as what was the origin of those viruses? What is the origin of life? Did they contribute to the life on Earth? Things like that. Do
they have large enough feature sizes that it's easier to study them using electron microscopy, or maybe not light microscopy? I don't know.
The point is, you're totally right. They are much easier to isolate because they are big. So you can actually see a viral particle by light microscopy, regular light microscopy. So that's very convenient because when you see that the cells are dying, you look at the culture medium, and by light microscopy, if the particles there are particles which are visible and with no motility, then you are one step in the direction of discovering a new virus.
So have you been able to observe them, entering? These are phages. They enter into bacteria. You said they're called Pandora viruses.
In the case of we have a unique cell system that we are using. It's a bias, we know that, but it's also a decision because it's much easier to work with a unique cell system. So those are amoeba, which are unicellular eukaryotes. And those viruses are actually infecting such amoeba. They all process, well, at least most of them do process exactly the same way, meaning that they are mistaken by the amoeba as food. They're the regular food of the amoeba, which is bacteria. And so they penetrate inside the cells through phagocytosis, meaning that they are actually engulfed by the cell, and that's where starts the infectious cycle.
Once they're engulfed, do they migrate towards the nucleus inside the amoeba, or does their capsid open up right away? Like, what behaviors have you observed?
For the 4 families of giant viruses that we have been isolated, they all remain in the phagosome, and there is there a signal which is actually opening the capsid, and you have a membrane which is inside the capsid, which is unfolding and fusing with the membrane of the phagosome. And so at that point, everything that was inside the particle is transferred inside the cytoplasm. This is the first step. Then the fate of what would happen depends on the infectious cycle of the different family of giant viruses. Some of them do not need the nucleus at all. And so because of that, they develop inside the cytoplasm what we call a viral factory. In the case of the ones that do use the nucleus, in one case, we were able to follow the journey of the DNA from the moment it was transferred inside the cytoplasm to the nucleus. And once it is inside the nucleus, you can see it unfolding and allowing the transcription machinery of the cell to start expressing the early genes of those nuclear viruses.
That's awesome. So you can you see the packaging of new virions by the amoeba cell?
Yes, we can follow that as well. I mean, you can spot every stage of the infectious cycle by electron microscopy, not by a regular microscopy. What you can see using light microscopy is actually we use a trick where we stain the cells we perform DAPI staining, which is staining nucleic acids. And because of that, we are able to really locate the viral factory, which is really enriched in nucleic acid as soon as the replication starts. And so because of that, we are able to see if the cells are all synchronously, sorry, it's difficult to say, infected. And in that case,
synchronously, okay.
Exactly, thank you. And so then we can follow, we can perform all the electron microscopy study of those cells also to have a deepest view and a more detailed view of the infectious cycle all along it.
This is really cool. This is a good idea to help study maybe all kinds of viruses. The lessons that you'll learn from doing this can apply in many places, I'm sure.
I hope so.
Well, can you fluorescently tag, parts of the virus or parts of the amoeba as well, and then with light microscopy, overcome the limit and see more of what's going on with that electron microscopy?
Yes, definitely. And there is one amazing case where we did a study using such approach, which is a case of, let's say, an intermediate in between cytoplasmic viruses and nuclear viruses. Those are the family of the Marseille viridae. They are much smaller, less complex than the giant viruses, but yet they are pretty complex with more than almost 400 genes. And those one we thought from the very beginning that they were also cytoplasmic because they encode the transcription machinery. And we saw that the viral factory was actually developing inside the cytoplasm. And because of that, we assumed they were cytoplasmic. But we used the trick, which is exactly the kind of trick you are suggesting. We overexpress nuclear proteins inside the nucleus of the amoeba to address the question whether those viruses were really truly cytoplasmic. And the reason why we did that is because when we analyzed the proteome of the virion, of the capsids, we found out that the transcription machinery was not inside the capsid. And because of that, we thought either they are using a completely different transcription machinery, which is inside the capsid, or they have to find a way to recruit the transcription machinery from the nucleus. And during the study of the infection, during the early stage of the infection, we were able to see the proteins which were inside the nucleus, those fluorescence proteins, we were able to see them leaving the nucleus to go to the viral factory. And as soon as about, let's say, 3 hours later, probably the transcription machinery was already produced, the virally encoded one. And at that stage, the nucleus went back to normal. And this is only the protein that left the nucleus. The DNA of the host was always confined inside the nucleus.
That's amazing.
Yeah, it is. And I think in terms of evolution, it's really very interesting because that suggests that the most likely is that viruses were fully independent originally from the cell nucleus, but became more and more dependent as soon as they were losing genes in a process which is common to parasites, which is they randomly lose some genes and at some point become more dependent from the cell. And witnessing such an event that you can have viruses that are intermediate in between cytoplasmic and nuclear is kind of a demonstration of the way they evolve.
Yeah, there's a lot of things you can do. This is interesting. Some things I thought of is, I guess I've never thought about it, but there's probably some, nucleus to cytoplasm signaling, stuff channeling back and forth. I know that happens, but I wonder what the signaling is. That's one thing. And then, if you have a bunch of amoeba and you infect, can you just infect one of them with this virus, or you have to just put a whole bunch in and they start spreading throughout the colony?
Yeah, the point is traditionally, when we start with an environmental sample, we never know how many viral particles are in there, and sometimes there are very few of them. So in that case, you have you will start with, let's say, one viral particle that will then propagate all along to the other cells. So very likely, at some point, you have one cell infected by one virus, and that will produce hundreds of new particles that will propagate the infection all around.
Well, since the Pandora virus is so big, can you just have one Pandora virus with one amoeba and see what happens? Okay.
Yes, we do that also, but actually, no, we do not do single cell analysis because we are not equipped to do such things. But what we do usually is we kind of clone the viruses by putting viruses in the culture medium and isolating one cell, many times one cell, to the point where we will have only one cell in a well. And later, we add fresh cells to look at the difference in between the different clones, if any, and to separate the population of viruses that could have been in the environmental sample originally. But now we wanted to do some single cell analysis, but that would not be possible by electron microscopy. As far as I know, it's not possible to do such things right now, because usually what we do is we perform inclusion. So we take a pellet of cells, and this is this pellet of cells that will be observed by electron microscopy. So you have many cells.
But since the virus and the amoeba are large enough to see under light microscopy, you probably won't learn the chemistry, but why not observe them, one amoeba, one virus macroscopically, observe it under light microscope and just see what you see?
But you can also do that if you have a population of cells. You can look at each one after another, and you can see actually the viruses not that easily when it's the start of the infection, where you have only one particle per cell. It's tricky, I would say, and not the enlargement is not enough, I would say. But you can actually mistake the virus for mitochondria, which is about the same size. So it's very difficult for that reason also. And once at the end of the infectious cycle, definitively you can see the population of viral particles which are inside the cells, and you can recognize them. I mean, it's very fascinating to see that. And you have then two different types of viruses, the ones that will explode the cell or lyse the cell, and you have the ones that will exit by exocytosis. And that is amazing to witness as well, because we have movie where we have recorded the exocytosis, where you can see the particle budding and going away from the cell. So as you said, because they are large, you can still catch some events which are amazing to watch.
Oh, so in some cases, instead of lysing the cell, the virions they'll become packaged in membranes, or they'll migrate to the edges of the amoeba cell?
Exactly.
Huh.
Yeah.
Yeah, it'll be interesting if, let's say you had a population of amoeba and you introduced the virus on one end, and then maybe you were able to to look at the amoeba on the other end on a time lapse, after a minute or 10 minutes, 20 minutes, and see if there's anything going on with the ones on the other side, if they're getting signaling from the ones that are being infected. It's just an idea. I don't know.
It's a very good question, actually. Some of our colleagues did actually study that. He produced an infection on amoeba cell, and we covered the culture medium, separated the culture medium from the cells, and then put this culture medium onto fresh cells that never saw the virus to study if it was a signaling in there. And interestingly, it appears that there is some signaling in between the cells. But in laboratory conditions, we work usually with very high MOI, multiplicity of infection, meaning many particles per cells, to warrant that the infection will continue, because we are interested in... we didn't do such thing. What we wanted to do, for example, is performing a transcriptomic study of the infection, so meaning extracting the mRNA from the cells, and then try to find out what was down-regulated or up-regulated in the case of the amoeba on one side, and on the other hand, look at the differentially expressed genes on the virus side, to be able to identify the early genes from the intermediate and the late ones.
Yeah, I was also picturing, what if you have just one amoeba cell with like 50, giant viruses around it? Does it think it's a feeding frenzy and it eats a whole bunch of them, and then it says, uh-oh, or does it eat one and then it stops, or do the the other viruses just bump up against its membrane and just sit there until it eats them. I mean, again, it's there's macroscopic behavior as well that might give you clues as to what you're doing too, what to look for.
Of course, and this is true. And it depends on the viruses we are working with. In some cases, like for the mimiviruses, it can happen that you have more than one particle that will be engulfed in the same times. And you can even have some independent events of engulfment from the same cell. So you can find at the end few viral particles inside a larger vacuole. But in most cases, it appears, and I would suspect, I'm working on such things also, but I would suspect that there is a signaling as soon as the virus penetrates for some viruses inside the cell, it changes immediately the parameter or the metabolism of the cell to stop it to ingest anything else. So if you put many viruses all together in the medium, then it can happen that many viruses will be engulfed in the same time by the same cell. But if you put much less viral material inside the culture medium, then the most likely would be for you to find one virus. And if you try to reinfect with another virus or another family of viruses, then there is kind of an exclusion phenomenon, which is probably initiated by the viral infection.
That's amazing.
Mm-hmm.
Is anyone in your lab, or are they studying the giant virus itself, like trying to infect it with other viruses?
We don't have to. That? Yeah, of course, we don't have to try, because it does happen that we isolate giant viruses with their associated virophages, and so there is in the case of the Mimiviridae, we performed a study which is fascinating, I think, in terms of ecology and what happened in the environment, because we not only those giant viruses can get infected by a virus, those, as I said, are virophages, but obviously you know about them, but they can also be found associated with a selfish DNA, which are about more like plasmid-like, but they are viral as well. And those are called transpoviron. So you can have a ménage à trois and even 4 between the cell, the giant virus, the virophage who can infect the viral factory, and the transpoviron, which is also replicated by the giant virus viral factory. But the transpoviron can be propagated by the giant virus inside the capsid and also by the virophage. So the virophage can help transmitting the transpoviron to other giant viruses.
Wait a minute. So the transpoviron, what did you call it? Transpoviron. Okay. Is it just a naked piece of DNA that is membrane enclosed, or does it have its own capsid, and when does it come out and where?
It is not a naked DNA in the sense that, yes, it doesn't have a capsid. It uses the capsid of the giant virus and of the virophage. But it is not totally naked. We did perform the proteomic study of the virophage and of the giant virus that were carrying each of them the same transpoviron. And you actually can see that inside the capsid, those DNA are associated with a specific protein, depending if they are loaded with the giant viruses or with the virophage. So there is really something specific in the association of the transpoviron with the vehicle he wants to use to propagate.
But does this only happen when the Pandora virus is inside an amoeba, or can it happen when it's in the virion stage? I guess they're not usually motile, so it's just all this happens only when it's inside a host, right?
Exactly, you're totally right. It only happens, those exchanges do exist in between the inside the host. They have to occur inside the host. And what is pretty interesting also is this is an article we just published at the end of 2019. You can actually perform competition experiments using different transpoviron inside the cell and see what happens. And what appears is when a giant virus is associated to a transpoviron, there is an exclusion phenomenon that will completely forbid the replication of the incoming transpoviron. Only the one which is associated to the giant virus will be replicated. But if you infect the cell with a giant virus that do not have any transpoviron and use a virophage to bring in the transpoviron, then this giant virus will become transpoviron positive.
Weird. Yeah, I don't know. This is crazy. Are there any other giant viruses that aren't phagocytosed but that fuse to the membrane of their host, or is this the only one we found?
I think in the case of the viruses of amoeba, they all use this specific entry. But in nature, you have other systems, like for example, in the case of unicellular cells that can be seen in the ocean, in Cafeteria roenbergensis, for example. It's a complicated name, but it's a cell. And this unicellular cell can get infected by a giant virus, which most likely transfer what is in the capsid using a system pretty close to the one of bacteriophage. And there is even another case, which are the chloroviruses, viruses infecting algae, which actually stick inside the membrane of the cell they want to penetrate in. They generate a change in the membrane that will swallow, if you increase the pressure, and so that way allow the genome to get inside the cell very fast. So you have other processes which are used, and probably there are many others that we didn't see yet. So you can imagine also that you could have membrane fusion at the external side of the cell, even if we never got that yet. To me, it seems a little bit complicated to fuse, much more complicated to fuse with the membrane of the phagosome than with the membrane of the cell. But maybe there is a good reason for that, which is probably that way the viruses are protected inside the vacuole. And also they need probably also a signal, and this signal is easily provided by the vacuole, which has specific condition in terms of pH, oxidative stress, and things like that.
Well, have you tried to take the virion when it's inside of a vacuole, or I forget what you call it when it's initially phagocytosed? Do you still call it a vacuole, or what do you call it? What is it in?
Yeah, it's a vacuole. We call it vacuole just because it's limited by a membrane, but at some point, phagosome is usually contains all the enzymes which are able to degrade what was inside, and obviously the particles are not degraded, so it's not exactly a phagosome, even if the initial process is probably phagocytosis, but it's just a matter of...
phagosome, okay.
Sorry.
Have you been able... Okay, so
we
have you tried to isolate... Once it's in a phagosome, isolate it right then and there before it gets out?
Yeah, well, the first thing we tried, and we actually, I am deeply convinced that if we are able to recover nucleoid, which is inside the virion that contains the DNA and probably also transcription machinery of mimivirus, for example, I'm pretty convinced that if putting that in an in vitro system would allow the replication of the virus, because it has many, many things except translation. So in the case of mimivirus, I wanted really to do that. And so I wanted we were never able to purify the vacuole, probably because we didn't try enough. But as a proof of principle, I wanted to get the amoeba fed by magnetic beads to be able then to recover the vacuoles just using a magnet. So we tried that using magnetotactile bacteria. The idea was the cell would feed on the bacteria. They are magnetic, so which means that using a magnet, once they are in the vacuole, we should be able to recover the vacuole and then analyze the composition of the vacuole to be able to reproduce an opening of the particles alone in a specific medium. But that failed. It's right now I don't have anybody. Yeah, I think so. And I think it could have worked. And there is, for example, such things have been performed for Dictyostelium, which is another amoeba, to study the proteomic composition of the vacuoles, of the phagosome exactly. Not the directly magnetic bits.
Are you able to create a Pandora virus virion, just the capsid part, and that's empty inside? Is that possible?
No.
Oh, really? Okay. I didn't know if you could isolate just the capsid part, because if you could, it would be interesting if you feed that to the amoebas and see what happens. There's nothing inside of it, or maybe it's the sequence is compromised, and then see what happens.
Yeah. The problem with those particles is they are very, very resistant, and almost the only way to open them up is to degrade them. So you have also to use proteases to be able to fragilize the capsid, and so you are never able to recover intact empty capsids. We were not yet able to.
But inside the phagosome, I mean, the capsid is opening. Is it being degraded inside there, or is it opening?
It is an opening, as I said. But
there's some instruction. There's some binding site. There's some receptor then on some part of the capsid that is being signaled, and then the thing opens up, like a Rubik's Cube or something.
I think it's more than one signal. So you have a pH factor, which is very important. The low pH is a triggering, but not alone. It is needed to open the capsid. A colleague also tried to use pH 2, high ionic strength, and also heating at 100 degrees, and that way he was able to open up the particles. But 100 degrees, I think, has a bad effect on the particle on one side. And but probably doing this, you could do what you propose to do, meaning that they open up, and so you can recover eventually the capsid and try to see what happens when you use those empty capsid to feed the amoeba.
Oh, go ahead, please.
There is also probably a proteolytic cleavage, pretty specific, that triggers a change in the opening of the particles. In the case of pitovirus, which is actually the one that has the most beautiful mouse of all the family of giant viruses, it's like a cork, but it has an hexagonal array that make it look like a honeycomb array, which is actually the cork closing the particle. And we saw actually that there is something clearly happening there. As soon as the particle gets into the vacuole, you can see the change of the cork. And for me, it's not only just chemical, it has to be some proteolytic signal as well.
Yeah, this is a whole different way of thinking about viruses. I mean, essentially they're like little locks. If you can figure out how to get them to open up before they enter a cell, I mean, then you don't have to block them. They'll just open up and then they're in trouble. Their contents will spill out. That's interesting.
True. Yeah, it is totally true. And if you are opening them up, it's over for them. Except, as I said, the fact that if you provide them with a ribosome in the case of the mimiviruses, I'm pretty sure that they will give you back new particles.
Yeah, that's why it'll be really cool to see the packaging of new virions too, how they're made, how the sausage is made, essentially. I don't know. I mean, everyone says they're made inside the cell, but I don't know if anyone's observed it.
Oh, yes, we did in very big detail. The case of the 4 families we have been working on, you can see how it starts. And for all of them, I mean, for each of them, they do use different way to synthesize the capsid and to package inside the capsid what is needed. So
what have you noticed?
So all of them do recycle the membrane from the cell to be able to build their own membrane, and this is central to the assembly as well. So the membrane is probably the seed of the assembly for the proteins that will make the capsid. And then you have different processes depending on in the case, for example, of virus, it starts with a kind of membrane vesicle on which the protein are starting to accumulate. And this one is making a spheric particle. In the case of Pandora viruses, it's an ovoid particle. It's like you have open membranes, and so you can see really the building of the particle from a tiny spot to the entire sphere being made, or to the entire ovoid being made. And same thing with the icosahedral viruses. You can see them budding in the periphery of the viral factory. You first recognize, let's say, a flat face, and the assembly progresses. It goes to the full icosahedral capsid being made, but still empty. And then you can look and see, because the nucleic acids are pretty dense, electron dense by electron microscopy, you can even see the DNA coming inside the capsid. And in the case of the mimiviruses, the last stage is really how they can get the fibril which are surrounding the capsid and making them look larger and give them their sweet taste that fool the amoebas that think that it's going to eat the bacterium. So you really can follow all those events. We don't have an answer on whom or which proteins are responsible for all of this. Some of them we know, some we don't know. But when looking at the assemblies is something you can do using those viruses.
Yeah, this is amazing. It's really cool. It makes a lot of sense for you to study these things because you'll be able to see and get a hint of what's going on in smaller versions or different versions of viruses.
This is true.
So what do you hope to figure out in the next few years with your research? Particularly, what are you looking at and want to discover?
I do think that there is a question which needs to be addressed on, and I started already, we started to work on that. We do think that those viruses could be eventually a reservoir to discover new metabolic pathways, which I think is very interesting, both in terms of fundamental knowledge and in terms of possible application. Because you know, as I do, as everybody does, what viruses are good at is hijacking the cell and taking the control of all the processes which are going on inside a cell. But so on the fundamental level, I do think that those viruses, different families of viruses are so different from each other that I doubt that they have a common origin. I do think that they did coexist all together. Their ancestors did coexist all together and coexisted with the cell, or what should become the cells at some point. And all those type of interaction taught viruses to take the best out of the cells they are infecting. So I do also think that at the beginning of life, many alternative strategies did coexist all together in terms of, let's say, metabolism or even nucleic acid building or enzyme from nucleic acid, ribosome, stuff like that. So you know all those debates about the origin of life, whom was first, metabolism first, or nucleic acid first, RNA word. My point is I don't think that the answer is one or the other. I do think that all those alternative premises did happen all together and were strategies that were in parallel and got to come in competition. And at some point, they learn to coexist and co-evolve. And every time a big step in evolution was to occur, it's because one system was able to adapt with the other system to produce something which was just much better. And I think the viruses are the loser of evolution in the sense that they were not able to perform as well as what was going to lead to LUCA. But they learn to communicate with the cell and to co-evolve with the cells and had no choice at some point to become parasite of the cells to take profit of those big changes that led to the cells. So because of that, I think that those metabolic pathways that we may discover may teach us some lesson on the things which are originally different in between viruses and cells.
Well, what's the best way for people to find out more? Do you have pictures or videos of any of the microscopy you've done? You know, amoebas eating the viruses blowing them open and stuff?
We have videos of phagocytosis? No, definitively not. We have electron microscopy images of all stages. There is also an article that was a review article on the 4 families of giant viruses. There are many articles that can be read on giant viruses, definitively, and review in particular that give some insight into some detail of their infectious cycle. But of course, I do have plenty of images. Usually they are published also, so they are accessible. And our articles are mostly open access articles, so which means that everybody can download them and read them.
Okay, I'll ask you to for some links that we can put in the show notes. But that's great. So Chantal, thanks for coming. And the stuff you're working on is really cool. Very interesting, and I appreciate you being here.
Okay, thank you.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.